integrate and understand molecular changes associated with immune response, stemness, and oncogenic processes, a pan-cancer study. So, Professor, you can start.
Grazie per l'invito. Volevo ringraziare tutti per questa organizzazione, l'opportunità di presentare la mia ricerca e in particolar modo le varie associazioni di ricercatori italiani nel mondo. I can switch in English. Can I present? Can I share my screen? I think we can. Can you see my screen? So. Okay. Hi, everybody. I'm Antonio Colaprico. I'm an associate scientist at University of Miami, in Miami, Florida. And today I will give you an introduction to some bioinformatic tools that we have developed for understanding, as you said, you can see the title, no? Molecular changes associated with immune response, stemness, oncogenic process with the pan-cancer study. I'm an engineer. My background is in a telecommunication engineer and a bachelor and master thesis and a PhD in bioinformatics from University of Sogno in Italy. And then I spent my postdoc in machine learning in Brussels, Belgium, and then I cancer bioinformatics postdoc here at University of Miami. What I'm talking today is particular NIH-NCI pan-cancer initiative that call it like TCGA, it's the Cancer Genome Atlas, and that call it also Genomic Data Commons. So those are large NIH-NCI initiative like the CNR in Italy, no? Where we can have a lot of several tumor types, let's say from different organ of origin, like brain, bone marrow, and breast, and we have a lot of cases, like 85,000 cases at different projects. So I think, I don't know, I skipped one slide. So my most important contribution to date in the cancer bioinformatics field has been my authorship and publication of TCGA links. It is an official bioconductor for this US National Institute of Health and also the National Cancer Institute. So let me talk about this TCGA. So it means the Cancer Genome Atlas. So it's, as I'm mentioning, it's an NIH large initiative where we collected, because I'm part of this TCGA consortium, 11 different patients from different 33 tumor types and 7 different data types. So it means that for the same patient, the same detect cancer, we can have multi-omics like gene expression, methylation, microRNA, protein, clinical data, and mutation. And there was a collection that we published in 2018 in Cell that was called pan-cancer Atlas, where there were like 27 papers. Particular TCGA links, we published like an NR paper in 2016. It's an RBA conductor package. We have also a Docker hub. I'm looking into the statistics. It's being used more and more, and we have a lot of citation. The idea when I vision had was, I wanted to have 3 things, collecting the data from cancer, and having querying the samples, downloading, preparing in a matrix, and then analyze them to show and visualize them to show some results. So for example, here, some functionality overview. When you have deletion, copy number, you can have amplification, deletion, you have mutation, you can have a non-complete plot, heat maps, and so on. Then we showed some cases like we integrated. I said integration, so it means using of 2 or more data types together, like we integrated for breast, colon, ganglioma, and kidney, like different tumors from our organs. We integrated like different gene expression, methylation, or gene expression, mutation, or survival. Because mainly the concept when we do cancer is that bioinformatics analysis is that we want to identify, for example, subgroups. We want to identify clusters, want to identify molecular subtypes that they can have differences, different features, so they can receive a different therapeutic options. One of them is this Carelli et al. In Cell. That was from my PhD former supervisor. And then we had, as I mentioned before, so we have multiple also angle that we want to touch the cancer. So immune system is important because cancer also is as an accelerating disease, a metabolic disorder, where we have also importance of the immune system. So what we did is that we discovered like 6 different immune subtypes with different survival outcomes here, no? And then we covered, interferon gamma dominant, TGF beta dominant. So those are also important for other, research studies. Then we study like cancer driver genes from different angle. We integrated 26 different software tools, and we came out with a new comprehensive cancer driver genes consensus with 299 genes. What we did was also using CPTAC data, and then using other tools, we developed this to identify, let's say, different survival outcomes in CPTAC samples where we had, not necessary survival data. So then the stemness. Sorry if I'm running, but so what is stemness? The concept that we wanted to understand if is the normal tumor, what let's say is the tumor or the normal adjacent tissues different as they engage normal stem cell phenotype to progress. So what we did was to design more complicated machine learning algorithm that we call one class logistic regression. We trained on some iPS stem cell, and then we derived some indexes, and then we used these indexes to do many things, to sort the samples. It's like the immune score, but in a different. So at the consortium, there is the International Humanity Genome Consortium. We took data from all these countries, so not only from Europe, but the American, and also South Korea and so on. They called International Humanity Genome Consortium. So for this, we have mainly a lot of epigenetic mechanisms, epigenetic derived H3K27, H3K27 acetylation, methylation, H3K phonomethylation, so histone marks, methylation, that histone. So it's important. I mean, because in the DNA we can have all these nucleotides that you can see here, all these. And then there is like chromatin, and there are specific histone markers that are important. I didn't report it here, but when we were studying aging, we observed like a total loss of H3K27 acetylation. So it means that as we age, there are some mechanisms that we need also to understand. What are cancer driver genes? Those are like drivers driving a car. It means that you can have oncogene or tumor suppressor. That oncogene, mutated genes that can lead to cancer progression. A tumor suppressor is a gene that can stop the tumor to progress. Consider that you don't have the brakes, so it's an oncogene. If you have the brakes, it can be tumor suppressor. So what we did, I wanted, to explain it. We collected 26 different algorithm in the literature, and then we defined this new list here in this paper. And but not only I designed and a vision and event that we made this Moonlight, this national communication. I made multiple bioinformatics workshops, tutorial, because I like the research should be open source, reproducible, understanding, free. And then we did like this poster and by conductors, but the national communication that I was mentioning, so was quite interesting, some interesting finding. Let's hide for the moment, how the background. But we were studying dual role genes. So it means some gene that can change between oncogene and became tumor suppressor depends from the tissue, it can be oncogene in breast and can become tumor suppressor in a lung. So when we were doing that, we found that we call it these oncogenic mediators exhibited differences in somatic and internal mutation. And also we found, so we confirmed one thing that was reported in this science paper that say, you know, there is tissue specificity in cancer. So there is epigenetic background responsible for the driver genes to change the role. So we confirmed open chromatin accessibility at the promoter region for oncogene, but we discovered open chromatin regions for intron regions for tumor suppressor. Sorry, okay, I think I have a few slides. So that one nice circle plot. So we wanted to show that, you know, color autorenal was, you know, colored by cancer type, breast, colon, and so on. And then in the ring there are in green oncogene, yellow tumor suppressor, and this line and red are the dual role. It means there are multiple genes because when they change the role is important, because if you're targeting an oncogene, you want to knock down, whatever you want to do. Like a CRISPR-Cas9 editing, you want to do like a mRNA editing, you want to design a drug. It's important if they change the role. We wanted to continue, and then we have this marker paper in TCGA where we used the Moonlight on the oncogenic process, and also Moonlight was able to pinpoint some mechanism associated with oncogenic process like ZES2, OCTA4, and SOX2. So are really important for the stems. One thing that I'm happy to share, that one last paper that I can show quickly was selected as a top 25 health science article in the world among 7,000.
Professor Antonio, excuse me, we are over time.
Okay, I have the last slide. Thanks. So with this paper that I think is really important, we discovered in TNBC, mesenchyma group, was lack of immune cells. And I thought, can we restore this? Can we transform immune code in immune opt and eventually suggest immunotherapy? And yes, so we inhibited the ZES2 and then targeting this PR2 polycomplex mesenchyma TNBC restored MHC class 1 and HLA, B, and C in TNBC mesenchyma model and 9 chemotherapy. That's my last slide. So I want to thank all the person from the Translation Statistical Bioinformatics Lab, person from the TCG Moonlight, all the scientific committee, and Association Time Research, and in particular also Fabio De Fure from SICA, and also Manuel Giorgi from Mexico, and also my family of origin, my dad, my mom and brother. In my family, I created like, you know, my sons and also my wife Isabella, because she was with me all this time and support and take care of the sons. And I was able to do all this research. The findings are reported here if you have a question. Thank you.
Thank you, Professor.